I see the target audience because based on the interest of the audience, I would like to structure my talk. So it appears more students. So probably I will only touch upon the basic framework for drug safety. Incidentally, I'm heading the drug safety division at the CDSCO for the country. So probably it was thought like a apt topic, and then they have given me this topic. I also thank the organizers for inviting me. Uh, Before going to the details of the drug safety or pharmacovigilance, so let us understand uh, how the drug development takes place, and what is the implications of this drug development and the data emerging from this development on the safety of the product. Sometimes the slides may refer to vaccines. So basically, the presentation was made keeping vaccine as a product, but ultimately the drug safety pertains to all, including vaccines also. Uh, as you know, so initially the drug molecules are developed, and then their efficacy is assessed and then taken forward. The safety is assessed from the non-clinical studies, and then certain data emerge what whether it can be applied to humans, and then phase 1 study, and then phase 2, phase 3 study, and then approval. So, but the number of patients which are included in each of these phases are small. For example, in case of phase 1, it may be scores of patients. In case of phase 2, maybe about 100 patients. And phase 3, maybe 1,000, a few thousand patients. And that is why the data that is emerging from these phases and the preclinical or non-clinical toxicity data is very small. And with this data, we cannot extrapolate completely to say that a drug is safe. For that matter, no drug is 100% safe to be used by humans. So, everyone has to expect some sort of adverse drug reactions or toxicity or tolerability, lack of tolerance. So, whatever that has been mentioned earlier, so a brief write up. I mean, it's a busy slide. Sorry for that. We will deal it in the future slides. As I said, the drug approval, and how do we assess the safety data? So at the time of approval, we assess the safety data from the phase 1 to 3 studies, and the uh, the assessment should be favorable risk benefit profile. That means, as I said earlier, there is always adverse drug reactions. There might be minor, there might be major, or in any way, but however, the efficacy should outweigh the risk. So, if only if the efficacy outweighs the risk, then the drug is approved. If you see, uh, out of the number of molecules, say 20 molecules in phase 2, phase 3, hardly one or two clear the hurdle and then they become approved as drugs. This is because during the studies, the safety profile emerges, and the safety and the risks are high when compared to the benefits. So these drugs were never approved by any of the regulatory agencies. And once the drug is approved, because the safety data available in humans is very small, so the safety data is followed up in the real world situation, and uh, regulatory actions are taken from time to time as the data emerges, and that is called the PBRER, periodic benefit risk evaluation report. So this every innovator, whoever places the drug in the market, has to assess the safety data from time to time, and they have to evaluate the benefit risk, prepare a report, and the report is examined by the regulator. Only if the benefit outweighs the risk, then it is continued to be marketed. Otherwise, the drug will be withdrawn from the market. So, what are the regulatory actions? Regulatory actions include the withdrawal of the market, and we have seen in the past many drugs have been withdrawn after they have been placed in the market. Like for example, COX2 inhibitors like rofecoxib, valdecoxib, they have been withdrawn, and in few other cases, in India we have withdrawn this product, but later on detailed study, so we have approved with a red black box warning, so that the patient is aware that there are additional safety signals, and only in cases where there are no other options, then this drug can be used. One such drug is pioglitazone, which is used, which is an anti-diabetic. So as I said earlier, the time for development from phase 1 to phase 3, that is the human clinical development, may take from 5 to 10 years. But during the COVID, what we have seen is that all these COVID vaccines, which have been approved from 12 to 18 months only, when compared to various vaccines from 10 to 15 years. So why it has taken such a long time earlier, and now why in a span of 1.5 years these vaccines were approved? That is because after each phase there is a follow up period. The patients are followed up for the adverse events, whether maybe after use or maybe after discontinuation. If the patient is getting any adverse events, or if they are serious adverse events, and if they are resolved by themselves, or where there is medical intervention is needed, all those things have to be followed, because of which a time maybe after each year, after each phase 1 to 2 years time is taken. But during the COVID, what happened, all phase 1, 2, 3 were done, but there was no follow up, because it was because it was emergency, and we urgently need to have vaccines, and that is why these vaccines were approved very quickly. And of course new technologies like mRNA technology and the vector technology, so they have been introduced, and because of that the launch has been made very quick. But we see now so many adverse events have been reported following the introduction of these vaccines, and number of deaths have been also been attributed, but causality has been attached to some of the vaccines, and in many cases no causality was linked to the vaccines. But however, ideal way is to have follow up, then only the drugs or vaccines are introduced in the market. So coming to the drug safety, so uh, we know the word about vigilance. Vigilance means to be cautious of any risk that we may face. So pharmacovigilance means be cautious of the adverse reactions of the drug, and that is called the drug safety. And what is the definition of pharmacovigilance? It is a science and activities towards the detection, assessment, understanding, and prevention of adverse effects of any or any other drug related problem. So all these activities are called pharmacovigilance. So first one is, it is science of detection, how you detect a adverse event, and how do you assess them, and how do you try to understand them, whether this particular adverse event is related to the drug or not, because many a time the patient will be using concomitant drugs, means more than one drug the patient may be using, or otherwise he is not a healthy person, he is a sick person, because of that he doesn't have the capacity to tolerate, and with all these reasons uh, will lead to the understanding of about the drug safety. And when you get a proper understanding, then we try to address the situation and prevent them. So this is the entire science of pharmacovigilance. So as I said earlier, the safety data emerges from all these preclinical phase 1 to 3, and phase 4 is the post marketing study. So after after human clinical development, the product is launched in the market, then comes the post marketing studies. These may be observational studies, focused pharmacovigilance studies, structured studies, case control studies, retrospective studies. All these things will lead to more safety insights about the drug, and regulatory action is taken. For example, in case of pioglitazone, because in the France they had a system, all the drugs were issued by the insurer. So they had a database of the users of the pioglitazone, and when they search the database, number of the users they experienced renal carcinoma. That means it was thought like there's a causality relationship between pioglitazone and the renal carcinoma, and because of that we try to take action based on that data. But Indian population is different, and not many cases were reported in India. So that's why we issued a black box warning with a caution that you should use strictly under the observation of the physician, and also monitor about the renal health. So then only this drug should be used, because pioglitazone is such a drug which is specific in that category, and there is no other
many options for that category of anti-diabetics. So, and safety data also emerges from the PSUR, periodic safety update report. PSUR is that as per the Indian law, when a new drug is launched in the market, the marketer has to submit the PSUR twice every year for first 2 years and then annually for the subsequent 2 years. So this safety data will be assessed by the regulator and they take regulatory action including updating of the prescribing information. So prescribing information lists out all the possible adverse drug adverse events that, for any patient, based on which whether it is a patient or with the physician can take a considered decision. And in case certain concerns are if there are safety concerns on any drug, then certain studies called PAS, post authorization safety studies, are mandated upon the marketing company by the regulator, so that to confirm whether the safety concerns are genuine or it is only because of any belief. So I touched this earlier also, clinical development. But what is the difference between the clinical development and the real world? In the clinical development, the patients are limited and they are under controlled conditions, and there will not be many concomitant illnesses will not be there. Other drugs are excluded. Long-term treatment data is not, generally it is acute treatment data only. And patients are monitored for adverse events. But in case of real world situation, hardly there is any monitoring, because doctor prescribes. After that, follow-up is very, very less until the patient reports. And then a number of broader range of patients. For example, in clinical trial, patients between 18 to 60 are enrolled. But in case of the real world, so right from the maybe adolescents, children to geriatrics, all categories of patients are enrolled. There are comorbidities, and events too rare also occur. For example, in case of say earlier drug was there, so analgin, so it used to cause blood cancer 1 in a million, but that cannot be detected in a pre-launch, otherwise in the human clinical development. But in the real world, then we come across such cases where it is causing blood dyscrasiasis. So that is why internationally that is withdrawn. So after approval, the new drug will be closely monitored and post-marketing assessment will be carried out, and then its benefit risk profile will be weighed. So there are various regulatory, in Indian context, Indian law, there are many regulatory requirements for carrying out pharmacovigilance. So we have the NDCT rules. Maybe students be aware that for new drugs or anything related to clinical development, there is something called New Drugs and Clinical Trial Rules 2019. These rules are made under the Drugs and Cosmetics Act. So under the NDCT rules, so whether manufacturer or importer has to have a PV system, pharmacovigilance system in place to collect, process, forwarding the ADRs to the licensing authority, because they collect the licensing, they collect the ADRs and they forward to the regulator. Regulator along with the manufacturer, they try to assess whether there is a causal relationship between the drug and the adverse event, and if it is there, then what is the way to address the situation, whether they are so serious, then the drug will be withdrawn, or whether the still benefit is there, then warnings are given. In that manner, actions are taken. So PV system shall be managed by qualified and trained personnel. So generally it is either the physician or the pharmacist who carries out the who manages the PV system. Post-marketing assessment. What are various steps in the post-marketing assessment? One is the phase 4 clinical trial, where the data is limited. Then we ask them to conduct phase 4 clinical trial, or when the drug is already approved in other country, but we don't have data in Indian population, even though we may approve the drug to be launched in the country, but we ask them to carry out this phase 4 clinical trial, so that the safety data is captured under a protocol. Protocol means it is structured study and all the data has to be submitted, and also the patient will get the medical management, compensation, everything is available. So this is the best option to have a study for carrying out safety data. But this is expensive and legally bound. So that is why they also carry out simultaneously other studies like post-marketing surveillance study or observational study or other interventional study for active surveillance. And also, as I said, PSUR reports data is submitted periodically to assess the safety. So coming to the PSUR, as I said, so PSUR, so they submitted for the first 4 years in India, but in other countries, so it is lifelong because the safety data is generally assessed by the captured and assessed by the innovator, not the generic manufacturers. But in India, everyone is a generic manufacturer, so that is why we don't have any company which carries out the PSUR until unless the manufacturer or importer launches the drug in India for the first time. And that's why we have a limited requirement of only 4 years, but in case of innovator, it will be for lifelong. So data will, all this data will be captured by the marketing authorization holder. PSUR report shall be submitted, and it pertains to all dosage form and indications because a drug have multiple indications, but the safety pertains to the same drug. But however, the Central Licensing Authority, means DCGI, can extend the period from 4 years to further for further period if he feels that the available safety data is limited or otherwise the patients which are on the drug are very small, so that no safety data emerges, then the duration of this can be extended. How the safety PSUR is carried out? The reports are submitted by the marketing authorization holder. Then we have expert committees with CDSCO, and after the data is after received, then we carry out the pharmacovigilance inspections, and it is confirmed whether the marketing authorization holder is confirming with the requirements of the pharmacovigilance system, meeting the pharmacovigilance system, and then under the expert committees will assess this data, and then they issue some recommendations. So recommendations may be referred to the signal panel. That what is signal panel will go through in the coming slides. So these are pharmacovigilance inspections because we short of time, so we'll skip this part of pharmacovigilance inspection. So objectives, they are routine inspections as well as targeted inspections, unannounced inspections. So process of methodology of pharmacovigilance inspections and reporting. This is a checklist. So what are the related documents that need to be done? So because PV is an emerging area, so in India, the domestic manufacturer does less pharmacovigilance because they are not innovators. But India being a what you call global house of the IT outsourcing, global IT outsourcing country. So the majority of the global PV systems, IT systems, are operated in India. All the major IT companies have a division of pharmacovigilance. So they capture their reports, they do the assessment, and they send to the principals whoever they employ. And that's why many pharmacists have employment with many of the IT companies and who work on this pharmacovigilance. These are the common findings and observations in the inspection reports. These are the classifications. I'm not going to details because of, so till date we have carried out 59 PV inspections, and then many other are under process because this division we have started very recently. So earlier it was carried out by other agencies. So coming to the signal. So what is a signal? Signal, what are the steps involved in signal detection? We will see in the coming slides. This is the definition of the signal. Signal is the information from one or multiple sources which suggest a new potential causal association. For example, if a drug is taken and we experience a adverse event, but whether this adverse event is related to the drug or not, we don't know. It may be that because of our health condition or maybe for some other reason that we might have experienced the adverse event. For example, even headache is an adverse event. A fever is a adverse event. So after taking any drug, if we experience any of these adverse event, whether it is related to the drug or not, we don't know. But we need to confirm whether it is related to that or not. And how do we confirm? We confirm when we receive more data, more such incidents of adverse event, and the drug. And once the adverse events are causally related with the drug, so this is done by experts by whatever means that I have shown you in the earlier slides. Event or set of related events that is just to be of sufficient likelihood to justify verificatory action. So everything here is statistics. So we always use, in case of vigilance, we always use these words, uh, likelihood, possible, probable, because it is difficult to confirmatory say that this drug will cause this particular adverse event. So this is the signal management process. This again, in case of vaccines, I have taken. So what are the adverse events reported? Minor, severe, serious, and then this database screen for what are the priority, of course, drugs or vaccines, and remove the duplicate case report, because it is possible that same report is received from multiple sources. The doctor sent the patient reports or the company manufacturer reports. So we remove the duplicate case reports, you apply the statistical methods, then both the qualitative review is there and also trend analysis is there, and present a signal assessment to the panel. So after submitting this data, the panel will assess the adverse, whether there is a causality relationship between the drug and the adverse event, and then it evaluates the benefit risk evaluation and gives a recommendation to the regulator. Regulator will take regulatory action. So there are three categories. So we know for 100% we know that there is no need for further evaluation based on the data. We are so sure, or in some cases we are not sure. So we need more data to confirm whether this adverse event is causally related, and ultimately, if there is a need for regulatory action, then the prescribing information is also updated, and safety restrictions are also issued by the regulator. So what does the signal review panel? This is the most technical panel. It consists of experts from pharmacology, clinicians, regulatory officials, public health officials, like that. These are the various steps: detection, validation, confirmation, analysis, prioritization. So this is the chain. So this is so maybe each one of us will have a part in any of this step. If you are patient, then we report. So if you work for the MAH holder, that is marketing authorization holder, then you collate, you assess, then you report, and then if it is a national PVPI program or WHO reporting center, then you validate, then confirm, and then if you are regulator, take final action, and then ultimately the manufacturer has to issue steps for risk minimization, because the benefit outweighs risk. That means the drug will continue to be in the market, but how to avoid the adverse events? So that is called the risk minimization. So this what the various steps. So ultimately it comes to the benefit risk assessment. So this is the signal management process in India. So who are the partners here? So the regulator, CDSCO. The state does not have any role in this regard. We have a pharmacovigilance program of India. There is a AEFI in case of vaccines. There is a under Indian Pharmacopoeia Commission, we have a pharmacovigilance program of India. So there are marketing authorization holders or manufacturers who report. So these are all the stakeholders who take part in the pharmacovigilance program. For example, this is regarding the Covaxin that we have seen during the COVID. Based on the adverse event data generated during routine immunization, Guillain-Barré syndrome is reported with unknown frequency. So this is the Guillain-Barré syndrome. Is something very, very fatal, lethal. So, but it occurs very rarely. We don't know about the frequency. But because of this entire process, then we have identified, because Covaxin is our own product. I mean, our own means Indian product, made in India product. So no one else in the world would report any safety issues with that, because majority of that vaccine has been used in India. It has been our responsibility to identify whether any causal relationship is there between the adverse events and the vaccine, and accordingly, so this has been reported by the AEFI and the CDSCO, and we have asked them to update this in the prescribing information. so that whether the physician, whoever reports, whoever prescribes to take this vaccine, or the patient takes a calculated risk. So this is the summary. So ultimately, so what I say is that spontaneous reporting is the most important thing. For example, as I said earlier, so majority of the drugs which are used in our country are generic drugs. So that is why there is no marketing authorization holder who carries out this burden of doing the pharmacovigilance studies for this particular drug. For example, paracetamol is there. It has been in the market for 50 years. Even today, there might be possibility that it might show new adverse events. We don't know, but who will come forward to capture these adverse events? Because there is no manufacturer who takes this responsibility because it is a generic drug. So we have the program called Pharmacovigilance Program of India. So under this PVPI, all the patients or the physicians, they send the spontaneous reports. Spontaneous reports means I have taken a medication, then I encounter some adverse event, then I report to the pharmacovigilance program. This pharmacovigilance program has adverse drug monitoring centers. So we have about 900 ADR monitoring centers in India, and more are coming up. So when we send the report to them, then they will assess based. They will ask for the history. So what are the concomitant medications that have been taken, and so what was the health condition? So whether this has been encountered earlier, all this information is captured, and they will try to establish a. They will say immediately one report they will not take any action. If we receive multiple reports of the same ADR for the same drug, then it creates a cause for action. So then it will be referred to the signal panel, and this signal panel will review whether there is a causal association between the even existing drug and the ADR. So once it is established, then we ask them to update the prescribing information. So as on date, PVPI, so we have identified 56 adverse drug reactions for the generic drugs which have not been reported by the innovator, because the number of patients who are using these drugs in our country is huge, because we have 1.4 billion people. So other countries don't have so many population, and patients are also more. So adverse reactions also will be more, and we are the ideal. We are in an ideal situation to capture these adverse events and then update them in the prescribing information, and also do the benefit risk assessment, and take regulatory actions. So ultimately, this is the spirit of this entire pharmacovigilance. So probably with this, I will end here. So thank you.
Speech on PV with all your inputs and insights. So we'll give 5 minutes time for the audience to have any questions.
Good evening, sir. So this in the case of various clinical studies, phase 1, phase 2, phase 3, why it is not possible to fix the number of volunteers for each phases to be observed? That is one reason. Because in
It's based on statistical analysis. So we can ask them to increase the number of patients, but that would not serve any purpose at this point of time when they do not know whether this particular molecule shows favorable efficacy as well as favorable safety. Then only it is approved. So without knowing that, no manufacturer, no company would like to invest so much more on by increasing the number of patients. No one does. Okay, so that is why it is only based on statistical analysis. So for example, what is the indication for which it is used, and whether it can be, it is objective or subjective based on various scales and the outcomes, then the drug is approved. So when we tell them, no, you instead of 1000 you do for 10,000, it becomes 10 times more expensive, and when they launch the product in the market, that will be so much expensive. So that is why it is not prudent to increase the number of patients at the time of phase 3, but subsequently they can follow up and have more in phase 4.
In a post marketing.
Yes.
So another thing is like this PB for pharmacovigilance. The thing is that this various drug combinations are coming.
Correct.
There actually there is no very synergetic action or subtherapeutic action. Whatever, for example, aceclofenac, serapeptidase, paracetamol. So many ranges, different categories are mixed together.
True.
What about the bio equivalence or bioavailability of each ingredient of that one?
Yes, see, thank you for asking this question. When a combination of existing drugs is made, then also it is called a new drug. New drug means then they have to come to the CDSCO, DCGI. They have to do all this. They need not to maybe phase 2, phase 1, phase 2, but they need to do phase 3. And once they show efficacy in phase 3, then they also need to do phase 4. In that at that moment also, all this safety data is captured. So we only to facilitate, because these are known molecules, we know at what strength we have so much information already in the public domain. For example, you said esomeprazole, serratiopeptidase. So we know we have a lot of safety and efficacy data on these molecules. So we directly go to phase 3, and when by combination, if they report any adverse reactions, so, that is captured in phase 3 and also phase 4.
Sir, why I am so particular about this one?
Yes.
For the last 40 years, I am in UAE only, Abu Dhabi only.
Yeah.
So I was actually trying to import so many such combinations from India to UAE.
Mhm.
So the UAE Ministry of Health
Mm.
was vehemently objecting that one, because saying that such a combination will not be allowed there because they don't have any
Yes.
biopharmaceutical
Correct.
background
Correct.
studies.
Correct.
So that is why I am so particular about that one.
So in India, because of our domestic compulsions and requirements, we have more number of combinations. That is because here compliance is less. For example, if you take the same case of esomeprazole and serratiopeptidase, if there is an peptic ulcer, then maybe after 2-3 doses itself, he'll get some relief, and then they'll stop using that, and that condition or that will recur again. So that is why, and the compliance more in case of fixed dose combinations. That is why when there is a rationality, these FDCs are approved. Thank you.
So it was a fantastic slide deck. I was very attracted to it. Maybe I will take it from you if it's permitted.
Absolutely.
Anyway,
Thank you.
my question is on patient safety rather than PV. Can you please comment on the status of ranitidine, sir?
No, the basic issue is that ranitidine has the nitrosamines. Though nitrosamines are cancer-causing agents, and these nitrosamines are not part of the ranitidine itself. They become during the manufacturing process they generate. But when we see the content of nitrosamine in each of the unit dosage, that is not that much to cause any concern. But if someone is using for extended duration of time, say years together, then there might be a concern about the safety, and there is a cause for maybe cancer. But there has been no studies, because such type of patients who have been using this drug for number of years and they reporting any ADRs or maybe cancers, so that data is not readily available. So that is why it has been put under watch, and the nitrosamine content has been monitored in not only in this thing, across all the for example sartans also nitrosamines are reported. And now whenever even the existing drug, say API, which is a generic, when application comes to us, we assess whether nitrosamine can be maybe during the synthesis they might be released or may be caused, and then if they are caused, then we are asking them to monitor what is the total amount of nitrosamines and whether they are within the limit or not. So this is the pathway that we are, but this is an emerging area at present. So it is only monitoring. We have not taken any action. Yes. I mean, depends upon their individual concerns.
Hello. What is procedure for reporting adverse drug reaction for
As I
patients?
said, no, they are AMCs, ADR monitoring centers. So ADR monitoring center.
Any
So if you,
online or something?
yes, everything is there. You can report to the ADR monitoring center. There are 900 throughout the country in all major medical colleges. Or otherwise, there is a pharmacovigilance program of India. There is a call center also. You can report to the call center. They will capture the information whatever you have, and they also come back to you to have more information to find out whether they can establish any causality with this drug. And this data is collated. We are also we are WHO collaborating centers for pharmacovigilance. That means we are the one of the largest contributor of ADRs to the WHO throughout the world. WHO has the collaborating center at Uppsala in Sweden. Then we contribute to them to further enhance the understanding about the drug safety and pharmacovigilance.
So we are running out of time, sir. So we consider this as the last question.
Only one short question.
Yes.
Why famotidine is not used today in this period as compared to omeprazole, pantoprazole?
I don't think famotidine is not prohibited. So it is only a choice by the see what happens. We are something called fad. Fad means fancy. So when we get a new molecule, all the physicians, everyone would like to go and prescribe the new molecule, even if it is 20 rupees, 30 rupees. Famotidine will be 25 paisa, 30 paisa. Still, we don't use famotidine because doctors are not prescribing. As simple as that.
Okay, so thank you all of you. Sir, most of the students are sitting here and they were listening your lecture very curiously.
Yeah.
It would be nice if you add something on phase 0 clinical trial.
Yes.
What is important and how and why it is being implemented.
Yes. So, actually, this phase 0, phase 1, phase 2, phase 3, there is no hard and fast rule that this starts here, this ends here. Everything is empirical. So, phase 1, the first time in humans, phase 2, dose ranging studies, phase 3, confirmatory trials. Phase 0 is one where the pharmacokinetics is being studied with the low dose to have a preliminary assessment whether we can proceed or proceed, because initially everyone is circumspect whether this drug toxicity is what is the estimation, we don't know. It might cause death, it may cause more toxicity, grievous hurt. So that's why, so we'll try to do phase 0 with the low dose and pharmacokinetics. We do study ADME. Based on that, if it is satisfactory, we proceed to the phase 1 like that. So these are different phases, and there is no hard and fast rule that 0 after 0 1 should be there. We can jump here, jump back, 2 and 4 happens, jump one phase depending upon the type of the drug, indication, and the data emerging, and also the satisfaction of the regulator.